Thank you and welcome again to our retinal research update. It's always a privilege and an honor to have you, the constituents who support the foundation, our friends, our volunteers, family members, on these events, whether they're live or online like today, to learn about the research. And again, we greatly appreciate the support you give the foundation to make all of this research possible. I'll be going through a few introductory slides, then talk about some gene therapies, genetic research, stem cell therapies, genetic testing, and my retina tracker, some other therapies. But I'm very excited about our panel today, which includes Doctors Chung, Dolgen, and Salam. And really, I look at my talk as sort of a warm-up for them. And I have some questions for them that I will ask, and then we will open it up for questions from you, the participants, who can send them in through chat or I guess over the phone, potentially. I forget how all those questions come through, but you'll be able to interact with the researchers. I know everybody being in isolation, a lot of you are spending a lot of time in front of your computers watching online TV shows and movies. And so to make you feel comfortable with this presentation, I want you to think of this as Retina Flicks. And today's show is a dramedy about researchers who fall in love with the retina. And the photo on the left here, in case you can't see it, is of Shannon Boy's lab. That's Shannon in the bottom right with her tongue sticking out, and that's a picture of the retina on the right. And these researchers are just so in love with the retina and the research, and they're trying to find treatments and cures for these conditions. So I hope you enjoy our installment of Retina Flicks. So anyway, we're at a very exciting juncture in the advancement of research, treatments and cures for inherited retinal diseases and age-related macular degeneration. As I often say, 10, 12 years ago, there were virtually no trials underway. Today, we're approaching 40. And again, that's for gene therapy, stem cells, some other approaches. And what's really exciting is thanks to the foundation and other commercial entities, there are many other promising therapies in the pipeline. And we estimate about 80. So not only are we excited about the things that are in clinical trials, but also those things that are poised to move into the clinical space over the next few years. Retinal diseases are very diverse. They affect people very differently. We're going to need a lot of different therapies to eradicate the entire spectrum of retinal diseases. So keeping the pipeline full is really important. And the foundation's role, we fund many different types of research, lab studies, translational research, clinical trials, career development awards, natural history studies, genetic testing. But a big focus for us is moving promising therapies out of the lab and into clinical trials. And when we do that, we can attract outside investments. And a lot of the commercial investments you'll be hearing about today, the commercial entities in our space, are investing because the foundation is getting research to the point where we can attract these outside investments. And our portfolio is currently about 80 projects around the world, the US, Europe, and other countries as well. So as always, I'd like to give a little refresher on the retina so you can understand better how these emerging therapies work. On the left, I have that proverbial side view of the retina blown up many, many times. The retina is the piece of tissue that lines the back of the eye. It's only a 0.5 millimeter thick. And it takes light that comes into your eye, and when it hits the back of the eye, it hits the retina. The retina takes it and converts it into electrical signals that are sent back to your brain, and your brain uses them to create the images that we see. So the retina is the light sensing tissue that makes vision possible. And toward the top of this sort of layer cake diagram are vertical cells that are the photoreceptors, the rods and cones. And remarkably, the human retina, the average human retina, has about 125 million photoreceptors. The colorful ones in this diagram, red, green, and blue, those are cones. Cones are concentrated in the center of the retina. They give us central vision. They enable us to read and recognize faces. They give us color vision and vision in lighted settings. So cones are really the most critical for many of our activities of daily living. Rods, on the other hand, are concentrated more on the periphery of the retina, and those are those long black cells. Again, they give us peripheral vision and night vision. And then both the rods and cones are nestled into this single layer of cells at the top of the diagram known as RPE cells. RPE cells are a support system for photoreceptors. They do waste management and they provide nutrition. And when those RPE cells are affected in conditions like Stargardt disease and other forms of macular degeneration, the rods and the cones will degenerate. So RPE cells are very important. Now on the right of the diagram, or of the slide, I have a picture of a rod cell that's in sort of a lavender, and a cone cell, which is a little shorter in green. And this just shows what the cell looks like. The bottom part of the cell is sort of the engine for the cell where all the proteins are made that help the cell function and sense light. And then the top of the cell is the sensory part of the cell. We call it the outer segment. And that's the part that actually absorbs light and makes vision possible. Okay, so let's move into gene therapy. As I'm hoping most of you know, inherited retinal diseases are caused by mutations in a single gene for most people. So the challenge is there are hundreds of genes, each of which, when mutated, can cause a retinal disease. But for most individuals, it's one gene that's the culprit. And gene replacement therapy is an approach where you're replacing that person's mutated genes, copies of that mutated gene, with normal copies to hopefully halt the disease. Now, gene therapy can be used to do other things, and we'll be talking about that. And there are other forms of genetic therapies. But for the first few slides, we're going to be talking about gene replacement therapy. And thus far, gene replacement therapy has worked really well in clinical trials. And that's why I say so far, so good. Number one, it's been safe. That's really paramount. And we've had signs of efficacy in a few of the clinical trials. And as I'll talk about in the next slide, we have our first FDA-approved gene therapy. Now, the way gene therapy works is researchers engineer viruses to carry the healthy, normal copy of the gene into the cells that need it, often the photoreceptors, maybe those RPE cells. Now, these viruses are safe. Any of the native DNA in the virus is taken out. You're putting in that retinal gene that you need, and the virus is very adept at penetrating the retinal cell so that genetic cargo can be delivered. And in this slide on the right, that colorful ball is the shell of the virus. We call that the capsid. And to keep things simple, you can think of the virus as like the delivery truck and the therapeutic gene as like the cargo. And we use these viruses because they are adept at penetrating cells. In our world, they're delivering good things and not the harmful things that unfortunately we're hearing so much about these days. So back in December of 2017, we were fortunate enough to have the first gene therapy approved by the FDA for the eye or any inherited condition. And that therapy is known as Luxturna, developed by Spark Therapeutics. And this particular gene therapy is for replacing people who have mutations in one specific gene, RPE65. And when that's mutated, it causes LCA, and in some cases, RP, LCA being labor congenital amaurosis. Now, we're proud at the foundation to talk about is that we funded about $10 million in research in early stages of this project at Children's Hospital of Philadelphia to move this emerging therapy into the clinical trial at Children's Hospital of Philadelphia. And was very exciting is early on, we had very impressive results, especially in younger kids. They were putting away their white canes. They were seeing the faces of their parents and their loved ones for the first time. Some kids could even see stars in the sky. And these kids with the RPE65 mutations had very advanced vision loss at a very young age. It's a very severe disease. So again, the results were dramatic. Children's Hospital of Philadelphia spun off a company called Spark Therapeutics to raise the money and get the therapy across the finish line, which again it did in December of 2017. And then earlier this year, Spark was acquired by Roche Pharmaceuticals. So now there is some really significant resources behind this technology, which we're pretty excited about. And one of the most important things to keep in mind about the success of what we now call Luxturna is that it's paving the way for development of gene therapies to treat many other inherited retinal diseases. The success of Luxturna is really opening the door for therapy development. And we now have somewhere in the neighborhood of 24 gene replacement trials now underway. And I'm going to cover a few of those in the next few slides. One of the companies in this space now is Biogen. And these two projects that I'll be talking about momentarily, choroideremia and X-linked RP, were started by a company called Nightstar, which was a spin-off of Oxford University in the UK. And then Biogen, I'm guessing it was a year and a half ago, purchased Nightstar. But Nightstar has clinical trials underway for both choroideremia and X-linked RP caused by RPGR mutations. In the choroideremia trial, they've moved into the last phase of that trial, which is phase 3. They've completed enrollment. In the earlier phase, they reported some encouraging results for efficacy. Visual acuity was maintained or improved for at least 1 year in 90% of participants. So hopefully they will continue to have those kind of results in phase 3. I'll note that the Foundation Fighting Blindness funded many years of choroideremia research in the UK, a gentleman named Miguel Seabra, to help get this and other choroideremia trials off the ground. X-linked RP, Nightstar is in a phase 2-3. This treatment has been safe as well, and they've reported some improvements, encouraging results for efficacy in terms of visual fields and retinal sensitivity at 6 months. This is still at a relatively early stage, so stay tuned for further results. And we're very excited about the progress that our partner Applied Genetics Technology Corporation has made, AGTC. You'll be hearing from Jill Dolgen, who is their patient advocacy lead when we get to the panel. But AGTC has been a partner of the foundation for many years. Researchers that we funded in the 90s and the early 2000s became their scientific founders. And they also have a clinical trial underway for X-linked RP caused by RPGR mutations. That's in a phase 1/2, and they've reported improvements in visual acuity and retinal sensitivity at 6 months. And we'll get a little more update from Jill on where that trial is at as well. They also have 2 trials underway for a condition called achromatopsia. The 2 major causes of achromatopsia are mutations in the gene CNGB3 and CNGA3. And in both studies, the treatment has been safe thus far, and they've had some encouraging results for efficacy. In achromatopsia, the overwhelming symptom is light sensitivity. It's sort of the opposite of RP. Most people with RP have night blindness. With achromatopsia, unfortunately, people have day blindness. Being in lighted conditions can be very uncomfortable, even painful. Poor visual acuity and poor visual color perception are also the hallmarks of achromatopsia. So in these early studies, they've had some participants who had improved light tolerance. So that's an encouraging sign in these achromatopsia trials. And we'll hear more about those from Jill when we get to the panel. Some other gene replacement trials that are underway, Spark Therapeutics has a choroideremia gene therapy trial, and Dan Chung, who is on our panel, can talk a little more about that. Sanofi has had 2 trials underway, one for Usher syndrome type 1B, one for Stargardt disease. Sanofi is trying to get out of the ophthalmology business, so they're actually looking for partners with these studies, so stay tuned. We're hopeful that those will get picked up and continued. There are also trials underway in Europe for RP gene therapies, PDE6 beta, RLBP1, and Harama in France, who is doing the PDE6 beta, just a few days ago announced that they're also pursuing a gene therapy for CRB1. That's still a ways from clinical trials, but when that gene is mutated, it causes LCA, and I believe it causes RP in some cases as well. So we're excited about that announcement. Shannon Boy, who you saw with her tongue sticking out in that early slide, has helped get a gene therapy for GUCY2D into a clinical trial. That's being led by Sanofi Genzyme. We funded work for her to make that possible. So we're very excited about that. Stay tuned for more information about that study. And then our partner MiraGTx, who is now owned by Janssen Pharmaceuticals, which is a subsidiary of Johnson & Johnson, has actually three trials underway, 2 for achromatopsia and 1 for X-linked RP, again, gene therapies, though they have not made any public announcements of results from those studies yet. So I meant to say this at the beginning, but one thing that I've done in the slides today in my presentation is I've embedded some frequently asked questions. It seems like we get a lot of the same questions every time we do a presentation. So I thought to mix things up and make things a little fun, or at least different, I would answer some frequently asked questions. So I don't remember if it's coming after this slide, but I know we're approaching one soon. So stay tuned for that. Alas, there is a frequently asked question. And patients and families always are asking, how long will a gene therapy last? And that's a good question. Gene therapy is really intended to be a one-time treatment that will last many years. We're hoping it'll last the lifetime of the patient. We know that, at least in the Luxturna trials, people have had the therapy for at least 10 to 12 years now. So we're hopeful that in many gene therapies that the efficacy will be sustained. But at the end of the day, the durability of a gene therapy will depend on not only the gene, but the stage of disease. It may be the case that for somebody with a really advanced disease, a gene therapy may work early on, but the durability may not be that great. The bottom line is we're still learning. So we're hoping one treatment will last a long time, and we'll learn more as our trials and treatments get further along in the time spectrum. So let's talk about, or I will talk about, some other approaches to correcting or genetic defects. And one has been in the news quite a bit over the past couple of years. It's gene editing. And the approach is called CRISPR-Cas9. And what CRISPR-Cas9 is, it's like a pair of molecular scissors that go into each cell, or most of the cells in the retina. They find the genetic defect and they snip it out. In some cases, they insert a new piece of DNA. That's part of the correction. But the bottom line is you can think of CRISPR-Cas9 as a smart pair of molecular scissors that finds the mutation in the gene and cuts it out. So unlike gene replacement, where you're just putting in a whole new gene to replace the bad gene, here you're doing editing. And what's nice about gene editing is that there are some genes that are big and difficult to deliver using gene replacement techniques, those viruses we were talking about. Where with gene editing, all you have to do is deliver the molecular scissors for the most part. So earlier this year, Editas and Allergan, in a partnership, launched the first gene editing project, CRISPR-Cas9, in the human body. And it happened to be for the retina, and that's in a phase 1/2 clinical trial. That first trial is targeting a specific mutation in the gene CEP290, which when mutated causes LCA. And they also have in their pipeline a CRISPR-Cas9 approach for USH2A caused by mutations in exon 13. Hopefully that'll get into the clinic before too long. The commonality between these two trials is that CEP290 and USH2A are big genes. They're difficult to deliver using those viruses that I was talking about. So gene editing is an interesting and promising alternative for those. And while the foundation isn't funding this project, we are funding about 5 different CRISPR-Cas9 projects. So we are heavily invested in this exciting approach. Now, another approach to correcting a genetic defect is called an antisense oligonucleotide, or an AON. And I'm going to try to simplify the concept here. I know that term is a little much for the tongue, antisense oligonucleotide. So there's a company in the Netherlands called Procure that's developing these AONs to address specific mutations, again, in LCA, that CEP290 mutation that I was talking about, as well as mutations in exon 13 of USH2A. But the way an AON works is it works like a little piece of tape or whiteout to mask the mutation in the genetic message. There's a sophisticated messaging system in your cells that carry information back and forth within your cell, so your cell knows how to properly use the genetic information to make proteins. Those messages are called RNA. And all that these AONs are little pieces of genetic tape to mask the mutations in the RNA. So again, phase 2-3 for LCA, they've reported vision improvements in their phase 1-2, so they moved into phase 2-3. For the phase 1-2 for USH2A, exon 13 mutations, they actually just reported some vision improvements early on for about 25% of patients, so that's encouraging. They also recently launched a clinical trial for P23H mutations in the gene rhodopsin. That just got underway. They haven't reported anything for that yet. And they're also developing AONs for a different mutation in USH2A, a mutation in ABCA4, which it causes Stargardt disease, and another form of LCA that they have not disclosed yet. So finally, to round out the gene and genetic therapy topics, I wanted to talk about optogenetics. And this is entirely different from gene replacement or gene correction. And on the right side of the diagram, I have that, or I'm sorry, on the right side of the slide, I have that retina diagram again. And in this case, the photoreceptors have this ominous black X marked over them. And this is to indicate somebody who has an advanced retinal disease and has lost all their photoreceptors. And that's what happens when somebody is completely blind. They've lost all their photoreceptors. Well, gene correction, gene replacement, those approaches aren't going to help these folks because they don't have any photoreceptors left to receive the replacement gene or the editing tool. But what researchers have found is that there is another type of cell in the retina called the ganglion cell that survives after photoreceptors are lost. And in this diagram, I have an oval around the ganglion cells. And what they've done in lab studies with animals is they've made those cells light sensitive, in essence, to operate something like photoreceptors. So it's a way to harness other cells. Optogenetics is a way to harness other cells in the retina for vision. And in this case, it's ganglion cells. What's nice about this approach is that it holds promise for people who have lost all their vision, and it's designed to work independent of the mutated gene causing the disease. And we now have 3 clinical trials that are underway for this approach. Allergan has one in the US, GenSight, a French company, has one in the UK, and then Bionic Sight, in partnership with AGTC, just launched a trial in Long Island, actually, for this approach, or they're in the process of launching that trial. A little, I guess, sobering or cautionary note about these approaches is we still don't know exactly what people will see. These approaches, each approach differs. Some use glasses or goggles or technology to help boost or amplify the signal. And we'll just have to wait and see how well these actually work. But the fact that they're in human studies is a very exciting and promising thing. And they not only hold promise for people with conditions like RP and Usher syndrome, but also AMD. So let's move into stem cells. We'll move out of the genetic therapies into stem cells. And I show in my slide here, I have a picture of stem cells. And actually on the right is what we call a 3D retina organoid, which is a retina-like structure built from stem cells. And that's really the exciting aspect of stem cells, is that you can use them to make virtually any cell type in the body, including retina, including photoreceptors or RPE cells. And from a single cell or a small batch of cells, you can make literally billions and billions of cells you need for therapies. And what's also exciting is that researchers like David Gam are now able to take a sample of skin or blood from a patient or a healthy donor, tweak those mature cells to become like stem cells, and then coax them forward to become retina. So mature humans can now be sources for stem cells. And don't worry, you don't have to take like a whole arm or leg. It's just a tiny sample of blood or skin to make all the stem cells you would need for literally thousands of therapies. And stem cells are being used in really two ways to treat retinal diseases, or at least in clinical trials. One is to protect the retina, and the other is to replace cells that have been lost. And a company called J-Site in Southern California has launched a stem cell trial. Actually, this happened a few years ago for a protection type paradigm. So this is to try to save cells that people still have. And it's targeting cones in particular. We call this neuroprotection. And the cells are stem cells that have almost fully developed into photoreceptors. These are injected into the vitreous, into the middle of the eye, and they're designed to release growth factors that help keep the photoreceptors, namely cones, alive. And in the phase 1/2 trial, they treated 28 patients, and at the highest dose, they saw that the treated eye performed better than the untreated eye. So that's good results. So now they've moved it into a phase 2B. They're studying this approach in 85 participants. These include people with RP, Usher syndrome, and I believe some other conditions. Now, a similar cell type is being used by a company called Retinal. They're in the UK, but they've launched a trial of their retinal progenitors here in the US at Mass Eye and Ear, and also a site in Phoenix. But instead of protecting or only protecting the retina, this is designed to replace cells that have been lost. So the hope is when these cells are injected underneath the retina, that they actually make connections with the existing retina and restore vision. In the phase 2 thus far, they've had a few patients that have had improvements of on average about 3 lines on an eye chart, and that's at 12 months. So that's encouraging results. The treatment overall has been safe, and they've shown these encouraging results. So hopefully we'll continue to see this kind of performance as the trial goes on. This again is for RP. I know there are Usher syndrome patients in the trial, and there may be some other conditions that are enrolled in the trial. Jason Commander, the gentleman on the right here, is somebody we've funded for a number of years, both with a Career Development Award and for related research. And we funded we provided a significant amount of funding for the lab studies that made this possible. So stay tuned for this. And then finally, there are a number of clinical trials for dry and wet AMD where the researchers are replacing those RPE cells that I talked about earlier in the presentation. RPE cells provide support to photoreceptors, but in the macular conditions like AMD, It's the RPE cells that are affected first. So these companies, and I have one here called Regenerative Patch Technologies, are coming up with RPE therapies. They're moving them into clinical trials, and the idea is to replace those lost or suffering RPE cells. Regenerative patch actually uses a scaffolding which mimics a very important membrane called Brooks membrane that helps provide an important barrier between the retina and retinal vasculature. So this trial has actually been underway for it's approaching 2 years. They reported some encouraging results early on. The most important, the patch, as they call it, was well tolerated, and there were some suggestions of efficacy. Now, this company is doing this for dry AMD. There are companies that are using this approach for both dry and wet. There's a group in London, they call it, I believe, the London Project to Cure Blindness, who has a similar approach, but for wet AMD. Okay, I think I have an FAQ coming up here. I often get the question, will doctors be able to transplant an entire eye? It's a very, it's something that we all would hope for, that we could replace an entire eye rather than worrying about therapies and transplants and things like that, or cell transplants. The answer is twofold. Researchers are working on it, but it's a very daunting challenge. And actually, a group that's been working on this for a while is at University of Pittsburgh Medical Center. There are a number of challenges in putting an entire new eye into a human being. One, they have to reconnect the vasculature, the blood vessels. Two, they have to reconnect the muscles, the musculature. And most challenging is reconnecting the optic nerve, which connects the retina to the back of the brain. That really thin cable, the optic nerve, has 1 million fibers in it. And reconnecting those 1 million fibers is really challenging. Now, I will also add that researchers are working on making entirely new retinas. And that may be something that down the road would be an approach to treating retinal diseases, not just replacing cells, but replacing whole new retinas. But again, you have that challenge of connecting the optic nerve. And to my understanding, we have a ways to go before that, that can happen. But there are researchers working on it. Okay, I want to talk about some other approaches to treating retinal diseases. And both of the approaches on this slide are cross-cutting, which is an important concept. Cross-cutting meaning they're designed to work independent of the mutated gene causing the disease. So you don't even have to know what a person's gene is necessarily for them to potentially get in a clinical trial and benefit from these approaches. One company in Dallas that we helped establish is called Nacuity. We're investing up to $7.5 million in their treatment. It's called NACA. It's a very strong antioxidant. Oxidative stress plays a major role in exacerbating vision loss in RP and AMD. And in lab studies at Hopkins, an earlier version of this treatment called NAC showed excellent promise in slowing retinal degeneration. NAC was modified and is now called NACA. I won't get into the details on that, but it was modified so it's more readily absorbed into the retina. There's actually a clinical trial of this approach underway in Australia, and we're working to get this trial off the ground in the US within the next year. Now, another company that we helped launch and we're investing up to 7 million euros in is called Sparing Vision. And many years ago, researchers in France at the Institut de la Vision discovered a protein that helps keep cones healthy and alive. and it's called rod-derived cone viability factor. And for those of you with RP, you may be aware that after your rods degenerate, rods are usually affected first in RP, then the cones go. The mutations that cause RP are actually in the rods, but after someone loses rods, because of the relationship between rods and cones, the cones will degenerate. And what the researchers in France discovered is this family of proteins called RDCVF, or rod-derived cone viability factors, that are secreted by rods that help keep cones alive. So this treatment is actually a form of gene therapy. It would be a one-time therapy, but the idea is you could try to preserve somebody's cones independent of the mutated gene causing their disease. And this, we hope, will move into a clinical trial within the next year or so. And then I wanted to mention a few Stargardt disease therapy clinical trials that are underway. And many of these target a similar or common pathway. So in Stargardt disease, the hallmark of the disease is the accumulation of these toxins underneath the retina in the RPE cells. And those toxins are caused by vitamin A processing. Vitamin A is necessary for the retina to be light sensitive. And your retina will metabolize vitamin A to make it light sensitive. But in that processing, there are these metabolites or these byproducts that can be toxic. In a normal retina, those toxins are cleared. But unfortunately, in people with Stargardt disease, those toxins accumulate in the RPE cells. The RPE degenerate, and then the photoreceptors also suffer. So there are a few companies that are targeting the vitamin A pathway. Alkeus is a company that has a modified form of vitamin A that people are taking in a trial that burns cleaner than standard vitamin A. They're in a phase 2. They haven't announced any results yet. Acusella and a company called B-Light, and there are actually other companies that are targeting vitamin A. Their approach is to slow the uptake or reduce the uptake of vitamin A into the retina. We call these visual cycle modulators. Acusella is in a phase 3. B-Light is moving into a phase 2 soon. I'm not going to go into the details about how these approaches work. They're a little different from each other, but again, the goal is to reduce the uptake of vitamin A. And then last, but by no means least, I wanted to mention a company called Iveric Bio, which is targeting a different pathway in the retina called the complement system. Researchers have found that the complement system, which is a part of the immune system, is overactive in people with many retinal diseases, including AMD, Stargardt disease, and others. It's like your immune system has gone a little haywire. It doesn't shut down properly. So Iveric has this protein called Zamora, which is designed to inhibit the complement system. That's in a phase 2. This approach is actually in a clinical trial. I believe it's still in a trial for dry AMD. But Iveric Bio has also gotten into the gene therapy business, and they're developing gene therapies for ABCA4, which is the gene associated with Stargardt disease, Best disease, as well as rhodopsin. And many of the approaches I talked about here may apply to dry AMD. Stargardt disease and dry AMD have a lot of common features. So a common or a frequently asked question that we get is, how long does a clinical trial take? And the answer, generally speaking, is 7 to 8 years, which I know can seem like a long time. We all want these therapies to move through trials more quickly. But researchers are really doing a number of things in these trials that are important. And really the thing that's most important is they want to ensure long-term safety. You want to make sure that the treatment is safe, and you can't just do that in 2 or 3 months. You do need a number of years to verify that something is safe. Researchers, especially at the early stages, are trying to figure out what the optimal dose of the therapy is. And this can take a few years to do. And then finally, at later stages of a trial, not only do you want to figure out that a treatment is safe and it's working, but you want different clinical centers to replicate the results. You want it to be something that many different doctors and clinicians can do. So this whole process, again, takes 7 to 8 years, but to ensure safety, ensure something works, you really do need that amount of time. And I know our panelists, all of them are experts in clinical trials, so you can save your further clinical trial questions for them. So now to kind of come around the home stretch here, I think I'm doing okay on time. We're going to talk about genetic testing. It's really important for everybody with an inherited retinal disease to get a genetic test. Number one, it confirms your diagnosis. A lot of different diseases can look alike. Once you find the gene, then you really have a sense for what the disease really is. Once you know that gene, you also know the inheritance pattern within the family, which other family members might be affected. You know better your prognosis, and you also will be able to qualify for more clinical trials. Many clinical trials will require that you know the mutated gene to participate in those trials. And with our program, the My Retina Tracker program, you can get no-cost genetic testing. The way this works is you go to your eye doctor, you tell your eye doctor, and it can be any eye doctor in the US, an optometrist, ophthalmologist, retina doctor, you ask them to order the test. They order it from Blueprint Genetics. It's ordered online at blueprintgenetics.com. And we have a lot of information on our website for you and your doctor so you can make this happen. And then it's Blueprint Genetics who runs the test and sends the results to your doctor. Blueprint has a really comprehensive panel. It's up to 322 genes, and it includes some of the more important genes that other panels might not include, including RPGR, which is associated with X-linked RP, and it can also find those harder to detect mutations called copy number variants and intronic mutations. These harder to find mutations are implicated in about 10 to 15% of cases. So it's really important that whatever panel is used to try to screen your gene, that you're looking for things like X-linked RP and these harder to find mutations, because they do contribute significantly to IRD incidence. Another important part of our program is your privacy is always protected. We never give away your personal information, sell it, give it away, whatever. We only share de-identified data, never personal information. Your doctor should get results in about 4 weeks. And as part of this program, you can get no-cost genetic counseling from Informed DNA. We require genetic counseling. Informed DNA is great. There's really no other company in the US, no other organization that has more knowledge in our space. And they provide an hour to 75 minutes of phone-based counseling. They have your report. They explain the results. They let you know what it means. In the case that your gene wasn't found, they'll help you with a path forward. They're really a good group. So we often get the question that doctors are having a difficult time giving a patient a diagnosis. Sometimes patients will say they go to one doctor, the doctor says cone rod dystrophy, another doctor will say Stargardt disease, Sometimes a doctor will diagnose somebody with choroideremia. Another may think it's X-linked RP. So the bottom line is the disease itself isn't as important as the, knowing what the gene is. When you identify the gene, you're really getting at the root of your condition, and that will tell you ultimately what the disease is and the best way to approach it. So the answer to this is don't worry so much about what doctors are telling you in terms of labeling the disease. Get genetically tested. Try to find that gene. That's what's really going to help you get to the bottom of your condition. And then finally, I wanted to talk about our patient registry, the MyRetina Tracker registry. It's on the internet. It's available to anyone who's been diagnosed with an inherited retinal disease. You go to myretinatracker.org. And by uploading your information into the registry, you're getting on the radar screen of researchers and clinicians from around the world who are doing studies and, importantly, developing therapies and moving those therapies into clinical trials. So it's a way to be on the radar screen, to raise your hand and say, here I am. You have to remember, these conditions collectively affect about 200,000 people in the US. They're all rare conditions. There isn't a lot of human data out there. A lot of patients aren't known to the research community. So it's important that you get into MyRetina Tracker to let the community know that you exist. And an important thing to keep in mind, people will go into MyRetina Tracker and say, you know, I haven't heard from anybody. It's been a year or a number of months. That doesn't mean that researchers haven't seen your record and don't know that you exist. Researchers are coming into the database and looking at the data, and they're planning for trials and therapy development for studies that may not occur for a few years. But being on the radar screen now lets them know that you're available for a trial that they may be launching at a later time. And again, researchers are using MyRetina Tracker to do clinical trial recruitment now. Again, your personal information is never shared. The researcher goes through this special process to get to notify us of their search results, and then we contact you, the patient, about the trial. The researcher doesn't know who you are until you contact them. So we have about 15,000 people in MyRetina Tracker. We've had more than 7,000 people as part of our program get free genetic testing. You don't have to be in MyRetina Tracker now to get free genetic testing. It used to be the case that you had to be, but that's no longer the case. And we've had about 150 companies and researchers come into Retina Tracker to look at the data, to do things like clinical trial recruitment. So we strongly encourage you to be in MyRetina Tracker. If you happen to not know the gene that's causing your disease, that's okay. You can still register. So one of the most common questions that we get, how do I know if there's a trial that's right for me? Well, as I just said, by getting into MyRetina Tracker, you can be notified about clinical trials for which you may qualify. MyRetina Tracker is not the only way. You can visit our website, look at the research section, and see which clinical trials are underway. We have a chart. We're writing articles all the time about trials that are being launched and ongoing. And then finally, at clinicaltrials.gov, you can get information on every clinical trial that's underway in the US and many overseas. And on clinicaltrials.gov, you'll get all the information, the inclusion, exclusion criteria, and also contact information for the trial sponsor, so you can reach out to them to let them know you're interested. And then finally, my last slide here, I'm doing a little shameless self-promotion here. I've been with the foundation for 15 years, and especially early in my career, I had the honor and the privilege to do human interest stories. And I interviewed a lot of families and kids who have inherited retinal diseases. And I've always been inspired by those families, especially the kids. You know, it tends to be the parents that are most upset about their condition. Not that kids don't get upset, but the kids are amazing. And over the years, they've really been my heroes. And I meet kids at seminars and the visions conferences. And I decided I wanted to write a story that made these kids heroes. And so in fall of last year, I had a book published called Retina Boy, which is in fact a sci-fi young adult novel that's a story about a kid born without retinas, and he becomes a hero. I'll just leave it at that. As I call it, it's a story of friends, aliens, retinas, and rock and roll. A little spoiler, as it turns out, he happens to be an alien from another planet. That adds a little intrigue and fun to the story. But he goes on to be a really excellent guitar player, and he has a band and a really smart girlfriend, and it's a lot of fun. So it's appropriate, I would say, for kids that are 12. There have been younger kids and their parents who are fine with it, but it's really a novel for people of all ages and abilities. It was published by Loyola University in Maryland. The publisher is called Apprentice House. It's on Amazon in paperback, Kindle, and Audible, but for a limited time, while I still have some copies available, I'm giving it away in Audible format and Kindle format. To get your free copy, email me, bshaberman@fightingblindness.org. And again, I don't have that many left, but I'll be happy to send you a copy. And if any of you out there have read the book and you enjoyed it, or even if you didn't enjoy it, I can always use reviews on Amazon and Goodreads. Okay. Again, great resources that you should access all the time: fightingblindness.org, clinicaltrials.org or .gov, and My Retina Tracker to upload your information. And I think we're right at an hour, so we can take a little breather. We can rest. I can take a drink of my water here.